Okay, hi, my name is Dr. Kara Yopak, and I'm an assistant professor and in the Department of Biology and Marine Biology at the University of North Carolina, Wilmington. As you know from what you're seeing on your screen, I run the UNCW Zombie Lab, which a lot of people ask me why on earth I would ever call my lab the Zombie Lab. And the main reason is that we focus primarily on understanding the brain. But actually not the brain you're more familiar with. We investigate the brain of fishes with a particular emphasis on the cartilaginous fishes. So that's going to be sharks, skates, rays, and chimeras. So really my lab focuses on three broad questions. So first we want to investigate how does the brain vary between species? And can looking at differences in the brain sort of allow us to make predictions about sensory specialization and behavior? And then we ask, all right, how does the brain vary throughout the life of an organism? And does that give us any indication of ecological shifts throughout ontogeny? And then particularly relevant to today's forum is, can we use the brain to try to predict the cognitive consequences of environmental change? So back when I was working on putting this together, I was sort of asked what gets you into shark brains. And ultimately, I have been fascinated with sharks since I was 5 years old. I was actually that kid. I was completely obsessed with learning everything that I could about sharks and always knew that I wanted to be an academic. So when I started my PhD, I really wanted to focus on shark behavior. But the more that I kind of dug into the field, I realized you can't understand behavior until you understand the brain. And therefore, a career was kind of born. Uh, in my experience, most people, when they think about working on sharks, they're probably picturing this guy. Of course, this is the white shark, star of stage and screen, tends to capture the attention of the media. Um, as do a few other very charismatic species like tiger shark, which has that characteristic striping down its body, or the bull shark, pretty common actually in our own backyard, can swim up freshwater estuaries. But we're really interested in the diversity of sharks. So I'm also just as interested in species that are smaller, that live near the seafloor, like this guy, which is the spiny dogfish, or the fastest swimming shark. That award actually goes to the short fin mako. Um, I particularly am interested in species that have kind of extreme lifestyles or morphological specializations, like the whale shark, actually the largest fish in the ocean, or the great hammerhead, which has that huge cephalofoil, as well as species that we still don't know a whole lot about, like this Greenland shark, who actually lives under the ice in the Arctic. So I think I could probably spend the next 10 minutes just showing you pictures of sharks and talking about why they're interesting. But the main point is we have over 1,200 species of cartilaginous fish. As you can imagine, just looking at this image, that encompasses a huge amount of diversity. So given this huge diversity, the big kind of first question that we're always interested in exploring is, are the brains of these animals similarly diverse? And how can we explain some of these variations? So to answer this, first thing you have to do is, of course, get out in the field. So my students and I spend a lot of time on boats in specimen collection. Although these days, mostly my students get to go out and do the work. Though, as you can see on your screens, occasionally they kindly will take me along. Um, to date, we've actually described the brain of over 180 species, and our current collection in the Zombie Lab is over 1,000 specimens across 50 orders of fishes. So we have quite a lot of tissue to work with and quite a lot of questions to explore. Um, in general, we use a really wide range of techniques to explore the brains of these animals. So that can range everywhere from normal dissection and histology, so sectioning the brain tissue. We also do a fair amount of scanning electron microscopy, which we take good advantage of the Richard Dilleman microscopy facility in Dobo Hall. We are also kind of at the forefront of applying advanced bioimaging techniques so that we can explore the brain in situ. So that is micro CT and MRI. And in addition to sort of allowing us to look inside these animals without having to cut them up, we can also generate 3D prints of aspects of their anatomy, which is incredibly effective as a teaching tool. So what are we talking about when I say that brains of these animals are diverse? I think there's this sort of common misconception that sharks are these pre-programmed, small-brained eating machines. But actually, there is a lot of work that's come out of my lab and that of my collaborators that's shown that sharks actually have relative brain sizes that are comparable to birds and mammals. And even more interesting is they were the first to have the full vertebrate brain blueprint. So they were the first to have a brain that's made up of all the major components that have then been conserved across all vertebrates, including you and me. But when most people think about a shark brain, and if any of you have ever seen one, chances are you're picturing what you see on your screen. So this is the brain of the spiny dogfish, Squalus acanthius. This is actually quite a common animal to dissect in a biology class, and is really what most people think about when they're considering overall anatomy of a shark brain. But if we were to only look at the brain of this animal, we really wouldn't be getting a sense of the diversity of brain anatomy. So as you can see from this slide, we've got considerable diversity that you do not need to be a neuroscientist in order to work out. So the spiny dogfish actually has what I would consider an average brain. So it's relatively small and nothing on it is particularly enlarged. If we compare that to something like this is the brain of the short-fin mako, and without going into too much of the details, essentially what's really large in a mako brain are the regions of the brain that process vision. So we can then infer that this is likely a very visual species, which we actually know to be true. This is an animal that lives in a very well-lit open ocean environment, and its brain very much reflects that. Different again from hammerhead. So hammerheads have among the largest brains of all of the sharks that we've looked at so far. Particularly notable is the region of the brain at the front here that is responsible for higher cognitive functions. So things like spatial learning and memory. We also tend to see enlargement of this structure in very social species. And in blue, as you can see, this structure, the cerebellum, also quite enlarged, and is a region generally associated with motor control and motor learning. So the big take home here is we see a huge amount of diversity in the brains of the animals we've looked at. Really what I'm putting in front of you are just sort of 3 examples, but if I were to put all 180 brains out on a table for you, you would start to put them in groups based on what looked similar. And what you'd find is the groups would be animals that are living in similar environments, swimming at similar speeds to catch similar kinds of prey, and have kind of a similar degree of social structure. So these kinds of questions, to me, are incredibly interesting, both from a brain evolutionary perspective and also in terms of questions on adaptation. So these huge variations we see in the brain really suggest to me that the relative importance of different sensory systems is highly variable across species. So if you were to look at the brain of a white shark, you would see really large regions associated with vision and olfaction or smell. If you were to compare that to the brain of a deep sea shark, suddenly those visual brain regions would be really small because they're living in deep, dark environments with very low light levels. But the regions of the brain that process non-visual senses would be very large. So we're starting to see this ecological puzzle in the brains of these animals that has huge implications for sensory specialization. And I think in addition to helping us understand their biology, when you understand biology and ecology, that goes a long way towards helping you with conservation efforts, as well as working towards developing more effective shark deterrence and limiting those shark-human interactions. So if that is one aspect of what we do. We have now this broad comparative data set, and something I think that's very powerful about that is we can now be predictive about behavior in species that are really difficult to study in the wild. So a recent study we did, published in Scientific Reports last year, we looked at the brain of the Greenland shark. This is a large-bodied shark, lives under the ice in the Arctic, and they are very surprised to find a very huge part of their diet are marine mammals or seals. And there was a lot of evidence that said they're being taken live. So these aren't being scavenged, they're not already dead when these sharks capture them. But these are very large, very slow swimming predators. And if you put accelerometers on them, they actually can't swim fast enough to catch these seals. So it wasn't clear what was going on and how they were able to do it. So in sending the brains to our lab, we were actually able to make some predictions about how they're doing it, which is that it's assumed that they might actually be following odor plumes to seals while they sleep, who are immobilized in the water. They sleep in the water to avoid being eaten by polar bears, but that actually might leave them vulnerable to these slow-moving cryptic predators in the water. So that's one aspect, is there's this huge variability in the brain across species. But the next question really for me is, okay, does the brain then change throughout the life of a single species? So when you and I are born, we pretty much have all the neurons we're going to have. We'll have a few zones where we can generate new neurons, but pretty much there's very little in the way of adult neurogenesis. Sharks are really interesting because they have a brain that grows throughout their life. So they're generating new neurons every day. So this lends itself to a very high degree of plasticity in the brain. So very recently, we just had a hot off the press publication, actually led by my honors student, Krista LaForest, who looked at this in one of our common coastal species, the sharp-nosed shark. And she was able to find that there are significant changes that happen in the brain throughout life, where the relative size of some of these major brain regions occupies different proportions depending on whether it's a juvenile occupying a coastal nursery or moving to more offshore habitats. So she suggested this may reflect a shift in sensory specialization throughout ontogeny, moving from an increased reliance on vision as a juvenile to an increased reliance on olfactory cues as that animal moves offshore. But we're not just shark specific. So these kinds of questions aren't just limited to sharks. We're actually exploring them in a number of bony fishes as well, including looking at how the brain changes throughout life in the sockeye salmon. So this has been the focus of my current master's student, who is looking at whether changes in the nervous system accompany lifestyle changes in this species as they move from freshwater to ocean environments and then back again to their natal stream. And we're particularly interested in how pollutants might be affecting normal development of the nervous system. So we have a lot of pollutants entering these freshwater ecosystems, and it's currently unknown whether they're going to negatively impact growth of sensory systems and brain, which is likely going to be critical for getting these animals back to their natal stream. So that's a little bit of a whirlwind tour through a lot of what we do, but it all kind of feeds into this final question, which I think is perhaps the most important of them all. So we see all of this variability in the brain across species that reflects an animal's ecology. And we see all this natural plasticity in the brain throughout their lifespan because they can generate new neurons so regularly. So to me, that begs the question, what degree do environmental conditions alter normal patterns of brain development? So this has been a huge focus of our lab going forward, particularly with some of the grim conditions these animals are facing in their environment. So as most of us know, as you saw earlier, our planet is experiencing an accelerated rate of warming. We've seen sea surface temperatures that have increased 0.5 degrees this century and are predicted to rise anywhere from 1 to 4 degrees by the year 2100. But we don't know the impact this is going to have on marine organisms, particularly ectothermic species like a shark, who actually can't internally regulate their body temperature. their body temperature is somewhat driven by their environment. So my PhD student at the moment is looking at these kinds of questions and asking whether increased temperature during rearing, so during early life, can actually negatively impact brain development. So we have 3 representative species that were reared either at ambient or 4 degrees above ambient conditions, which are predicted to be potentially the ocean conditions at 2100. I really wanted to keep the presentation data light, but I thought the results were still impactful despite being somewhat preliminary at this stage. So preliminarily, we are definitely seeing a reduction in brain size in animals reared at higher temperatures, but they are not homogeneous across the whole brain. So specifically, we're seeing significant reductions in size of the brain region for odor processing, as well as the regions of the brain associated with higher cognitive function, like spatial learning and memory, though we're not yet seeing differences in any other region of the brain. But as you might imagine, this has a lot of implications for negatively impacting how these animals are going to be able to detect and process odor cues, which is pretty important if you're a shark, as well as some of those higher cognitive functions, such as essentially learning where you live, learning where to go, learning where food patches are, as well as, very importantly, nursery grounds. So we're currently working to push these questions even further. In addition to continuing to understand changes to the anatomy, we really want to see if these anatomical changes also predict underlying behavioral changes as well. So we're planning to test for olfactory tracking to see if that is impaired in animals reared at these higher temperatures, as well as lower pH conditions. And then we're planning to do some cognitive testing in these sharks as well to see if their learning capabilities are similarly negatively impacted at these future ocean conditions. And unfortunately, these are showing reductions in the brain are really showing a predicted impact on their ability to perceive critical sensory cues as well as process those cues, and might suggest sensory and cognitive consequences for these changing ocean conditions, which is going to be pretty important for survivorship of a number of these species. We're looking at several different species of shark as well as ray and skate, and we're seeing some differences across species, so some may be more resilient than others. So I always like to kind of end the way I began, going from that 5-year-old who always wanted to study sharks till now. Safe to say, I think I've come quite a long way from this day, but I think probably more importantly, we have a really long way to go. So I'm very excited to see where the lab is going to go next and what kinds of questions we're going to be able to answer in the next few years. Thank you.